Antibodies are made of several different peptide chains, bound together in a Y shape. An antibody is made of 2 identical immunoglobulin heavy chains and 2 identical immunoglobulin light chains. Roughly speaking, the structural features of an antibody can be split into 2 parts: the domains that recognize a target, known as the variable region, and the domains that determine the antibody's biological function, known as the constant region. The heavy chain constant region of an antibody determines where in the body the antibodies are located, what kind of immunological response an antibody is able to mediate, and what the oligomeric state of the antibody is. Carbohydrate chains are attached to the heavy chain constant region and sit between the 2 heavy chains. They are associated with stability and effector function of the antibody. Heavy chain constant regions also determine the class of the antibody, known as the isotype. Different isotypes can have different oligomeric states, with immunoglobulin A, or IgA, forming a dimer, IgD, IgE, and IgG forming monomers, and IgM forming a pentamer. The 2 heavy and 2 light chains of an antibody are held together through disulfide bonds within the constant regions. A set of 2 disulfide bonds connects the 2 heavy chains in the hinge region, while the heavy and light chains are bound together with a disulfide bond that bridges opposing cysteine residues within each constant region. A second way of classifying the structural regions of antibodies is based on how antibodies can be cleaved by proteolytic enzymes. The enzyme papain cleaves just above the disulfide bonds within the hinge region, leading to a constant region or FC fragment and 2 antibody binding or Fab fragments. Another proteolytic enzyme, pepsin, cuts just below the disulfide bonds within the hinge region, which gives a single Fab2 fragment, with 2 arms still connected. Generating these separate antibody fragments in the lab has enabled the function of each region to be determined, while each antibody fragment can be useful for a variety of biological assays. An antibody binds its epitope through the variable regions in the tips of the heavy and light chains. There is an enormous amount of diversity in the variable regions, so that different antibodies can recognize many different target epitopes. Each of these chains contains 3 complementarity determining regions, or CDRs, located at the tips of each variable domain. Parts leading up to those tips are called the framework region. Most of the diversity between different antibodies is generated within the CDRs. Together, the 3 CDRs form the epitope binding site and determine the specificity of individual antibodies.